Welcome to my YouTube channel. Hello guys, welcome back. So here is my new lecture and the topic is spermatogenesis.
Spermatogenesis, as you see here, what does this mean? So the spermatogenesis means the formation of sperms. Genesis means formation and sperms की formation को क्या बोलते हैं? Spermatogenesis बोलते हैं. So spermatogenesis is a process जो कि मेल्स के अंदर देखने को मिलता है, ऐसा प्रोसेस जहाँ पर स्पर्म फॉर्म होते हैं. सो आई एम टॉकिंग अबाउट द मेल रिप्रोडक्टिव सिस्टम वेयर द स्पर्म्स फॉर्म. The primary sex organ in male is the testis. You can see here testis, which produce gamete called sperm. So testis produce the gamete called sperm. एंड अगर मैं टेस्टिस की बात करूँ तो टेस्टिस में एक सेल्स होते हैं. दीज सेल्स आर इमेच्योर मेल जर्म सेल एंड दीज इमेच्योर मेल जर्म सेल्स आर कॉल्ड स्पर्मेटोगोनिया, जो कि आपके क्या करते हैं, एट द टाइम ऑफ प्यूबर्टी दे प्रोड्यूस स्पर्म्स. सो दिस होल प्रोसेस इज नोन एज स्पर्मेटोजेनेसिस, वेयर इन योर टेस्टिस द immature male germ cells, that is the spermatogonia, it produces sperm, and the production of sperm and the formation of sperm is known as the spermatogenesis, and basically it starts at puberty. So if I'm talking about spermatogenesis, so it occurs in 4 stages. As you see here, the 4 stages, one is the spermatocytogenesis, meiosis 1, meiosis 2, and the spermiogenesis. So these 4 stages occur in spermatogenesis. First step is spermatocytogenesis, and in this your immature male germ cell which is present in testis known as spermatogonia. So these spermatogonia or immature male germ cells are present on inside wall of seminiferous tubules of testis. So they undergo mitotic division, that is also known as equational division. As spermatogonia are diploid in nature and contains 46 chromosomes, so after mitosis the progeny also have the same number of chromosomes, that is diploid and contain 46 chromosomes. So these spermatogonia divides and divides and divides, and they will increase their number. So lot of spermatogonias are formed, but some of the spermatogonia which form here undergo changes. So they grow in size like this and increase in size by taking nourishing materials, and those spermatogonia which undergo changes, they become the primary spermatocytes. So this whole step is the first step of spermatogenesis, that is known as spermatocytogenesis. So the second stage of spermatogenesis is meiosis 1. Here you can see that primary spermatocyte which is formed in stage 1. It is diploid and contains 46 chromosomes. It undergo meiosis 1. It completes its first meiotic division, which is also known as reduction division, and form 2 equal haploid cells which are known as secondary spermatocytes. So the third stage of spermatogenesis is meiosis 2. Here you can see that the secondary spermatocyte which is formed at meiosis 1 stage, which are haploid in nature and contains 23 chromosomes, they undergo and they complete their meiotic second division. And meiosis is also known as reduction division. So you see that here there are 4 haploid cells formed, and these 4 haploid cells are known as spermatids. So the fourth step is the spermiogenesis. As you see here, the transformation of spermatids into sperms or spermatozoa is known as spermiogenesis. So here you can see that these spermatids are non-motile because they do not have any of the locomotory structure and they are heavy, but when they convert into sperms or spermatozoa, their weight is reduced to half. Along with that, locomotory structure is also present, as you will see in the sperms or spermatozoa. Spermatids has organelles like mitochondria, Golgi body, centrioles, etc. Remember, this line is very important at the point of view of your entrance examination. So the Golgi complex of spermatids give rise to the acrosome of a sperm. So when we gonna study about sperm structure, then I will told you that what is this acrosome function. It is very important. It plays a very important function and role in fertilization process. So this is the structure of seminiferous tubule as you see here. So after spermiogenesis, what will happen? You can see here spermatogonias, okay, the immature male germ cell. These are the primary spermatocyte, these are the secondary spermatocyte. This is the Sertoli cells in which spermatid is embedded. So the spermiogenesis means when spermatid becomes sperms. So when the sperms goes out from the seminiferous tubule, they are happy, and this process is called spermiation. So when they are released out, they are happy. You will see here they are very happy because they are moving out from this seminiferous tubules. So this process of moving out of the sperm from the seminiferous tubule is known as spermiation. We study about hormonal control of male reproductive system. So at the time of puberty, there is increase in secretion of this gonadotrophin releasing hormone by hypothalamus. It is a part of brain, and due to increase in secretion of gonadotrophin releasing hormone, it acts on anterior pituitary, and anterior pituitary starts releasing two hormones, the FSH and LH, the follicular stimulating hormone and the luteinizing hormone. This follicular stimulating hormone has S, and the Sertoli cells also have S. So FSH acts on the Sertoli cells, and LH, that is the luteinizing hormone, acts on Leydig cells. The LL is same. So LH, that is luteinizing hormone, acts on the Leydig cells, and FSH acts on the Sertoli cells. Sertoli cells and Leydig cells are present in seminiferous tubules of the testes, and luteinizing hormone acts on the Leydig cells of the testes, and Leydig cells start secreting androgen, that is a testosterone hormone responsible for spermatogenesis, that is sperm formation. Similarly, FSH, that is follicular stimulating hormone, acts on the Sertoli cells of the testes, and Sertoli cells helps in the production of sperms, that is spermatogenesis. And also it releases a secretion of special protein hormone known as inhibin, which is released, that is inhibin, which suppress FSH synthesis by anterior pituitary. So when there is an excess production of sperms, then this cells, that is Sertoli cells and Leydig cells of the testis, start producing a negative feedback and stop the anterior pituitary to release FSH and LH, and also it decreases the level of gonadotrophin releasing hormone so as to stop the process of spermatogenesis. So the structure of mature sperm. The human sperm was first seen by Ham and Leeuwenhoek. So we're gonna talk about now structure of mature sperm. So it has four parts. You will see here the head, neck, middle piece and tail. So in the diagram I will show you, this is head portion, this is the middle piece, this is the neck area, and this is the tail. Above the head portion, anterior portion of the head is called acrosome. This area, this crown-like area is called acrosome, and you will see a membrane over the whole of the sperm, and this membrane is called the plasma membrane. So they are the four structures of the four parts of the sperm. Head, and it has a very little cytoplasm and contains haploid nucleus, that is 23 number of chromosome present in the nucleus. Here you can see the anterior portion is cap-like structure which is known as acrosome. So what is the acrosome? So acrosome is basically filled with enzymes known as sperm lysin. It basically dissolves the membrane envelope of the ovum. So when sperm reaches the ovum, this acrosome layer helps in dissolving the ovum's envelope and help the sperm cell to enter the ovum. So basically it helps in the fertilization of ovum. Sperm is derived from the Golgi apparatus. Second structure is neck, which is short and contain two granules, the proximal centrioles and distal centrioles. So basically this proximal centrioles help in plays a very crucial role during the first cleavage of the fertilized ovum. So when the ovum fertilized, that is the zygote forms. So after the zygote, there is a starting of division of the zygote. That first mitotic division of zygote, this proximal centriole will help. Now the distal centriole, what does this do? So this distal centriole plays a very important role and it gives rise to the long filament of the tail. So the long filament of the tail arise from the distal centrioles of the short neck. So the third structure is the middle piece, and it contains mitochondria which are 20 to 30 spirally arranged, and middle piece is also known as the powerhouse of the sperm, as mitochondria provides energy to the sperm for sperm motility or the sperm movement essential for the fertilization process. So fourth structure is tail, as you will see here. So basically tail is surrounded by small amount of cytoplasm and a cell membrane. So cell membrane is present and cytoplasm, a little bit of cytoplasm is present. Sperm swimming rate is 1.5 to 3 mm per second. So within 30 minutes they reach the site of fertilization. So they reach and fuses with the ovum and fertilization happens. So human male ejaculates 200 to 300 million sperms during coitus, and for normal fertility at least 60% of the sperms have normal shape and size, and out of 6%, 40% have the vigorous motility. So this is all about the structure of sperms we studied.
Thanks for watching.
Guys, if you like this video, please subscribe, share, and of course like this video. Thank you so much.